Exploring the deep marine biosphere, challenges, innovations, and opportunities. The deep marine biosphere is one of the largest and yet least explored microbial habitats on the planet. Quantifying the extent, diversity, and activity of subsurface microbial communities is a crucial part of understanding their role in global biogeochemical cycles. Even though deep biosphere habitats can vary widely in chemistry, temperature, turnover rates, and energy sources, all subsurface microbes inherently experience high pressures. While not all subsurface microbes require elevated pressures, for many high pressures are essential to their cellular function and metabolism. Thus, when targeting this elusive portion of the biosphere, it is critical to maintain in situ pressure while sampling and cultivating subsurface microorganisms. In this perspective paper, we highlight the sampling and cultivation technologies available to study these communities under in situ conditions. Maintaining elevated pressures throughout sampling, transfer, cultivation, and isolation is challenging, and more often than not, samples are decompressed at some point during sample handling, potentially leading to biases in both community diversity and isolate physiology. The development of devices that maintain in situ pressures during sampling and allow for sample transfer without decompression have begun to address the challenge, like the PUSH, Pressure Underwater Sample Handler. Such vessels can be used for both retrieval and enrichment of deep subsurface samples, as well as high-pressure growth and physiology experiments, thus expanding possibilities for deep biosphere exploration. Finally, we discuss the significant need to develop and share high-pressure facilities across the deep biosphere community in order to expand the opportunities to discover novel piezophiles from the deep subsurface. Unveiling the invisible majority, the deep marine biosphere. In this communication, we consider the deep biosphere to include all environments at and above 10 megapascals in the water column, 10 megapascal per kilometer. Subsea floor sediments, 15 megapascals per kilometer, and all potential reservoirs in the continental and oceanic crust, 25 megapascals per kilometer. Though we focus primarily on marine environments. The deep sea, defined as waters and sediments beneath 1,000 meters depth, is the largest ecosystem, representing 65% of the planet's surface and encompassing 88% of the global biosphere. The ocean has an average depth of 3,800 meters and an average pressure of 38 megapascals, and even the deepest waters are known to host life. The habitable zones of the continental crust are similarly vast and underexplored, and also host unique microbial communities. This remote and dark biosphere hosts a range of subsurface habitats typically characterized by extreme conditions. They are mostly nutrient poor, excluding the hotspots, e.g. hydrothermal vents, and have slow or transient energy fluxes supplied by water-rock reactions and circulating crustal fluids. Despite harsh conditions, these ecosystems define the largest microbial habitat on Earth. The subsurface is estimated to hold roughly 15% of the total terrestrial biomass, and resident microbial communities potentially play an important role in mediating global biogeochemical cycles. However, only 5% of the oceans have been investigated using remote instruments, and less than 0.01% have been sampled and studied. Despite being such a large fraction of Earth's biomass, the microbial diversity, activity, and distribution, metabolic pathways, and energy fluxes of the subsurface biosphere are poorly understood because access is limited by technical and economic challenges. Over the past decade, international drilling programs, IODP, International Ocean Discovery Program, ICDP, International Continental Scientific Drilling Program, as well as national submersible facilities, have expanded subsurface microbiology research to better understand how intraterrestrial life inhabiting the deep crust and deep ocean play a role within the deep biosphere. Subsurface technologies and recent advances in this field are well documented in Schwenk et al. 2010, Edwards et al. 2011, and Edwards et al. 2012. A majority of prokaryotes live under high-pressure conditions, and thus pressure is a governing factor for the distribution of life. And subsurface microorganisms are classified by their physiological responses to pressure. Usually, well-adapted surface microorganisms are negatively impacted by high-pressure conditions, piezosensitive, even though some can grow at elevated pressures, piezotolerant. Likewise, microorganisms isolated from the subsurface often grow optimally at high pressures, piezophiles, and obligate piezophiles are unable to grow at ambient surface pressures. In the case of the deep marine biosphere, evidence suggests that decompression can deleteriously impact cellular viability, including morphological changes, membrane cell rupture, and piezophile inactivation. Furthermore, significant shifts in community composition and gene expression have been noted as a result of sample decompression. To date, the number of facultative and obligate piezophiles is limited to roughly 56. Among the obligate piezophiles, Coluwilia marinemanae has the highest optimum growth pressure at 120 megapascals. This psychrophile and the hyperthermophilic Pyrococcus yayanosii currently claim the highest pressure limits on growth, 140 and 120 megapascals respectively. Although the majority of currently known piezophiles were isolated following decompression, these isolates may be notable exceptions representing only the fraction of the subsurface biosphere tolerant of decompression. Indeed, a much broader microbial community has been identified in the subsurface through alternative methods, including genetic and molecular approaches, in situ metabolic rate measurements, intact polar lipid analysis, stable isotope enrichments, cell counts, and growth of viable cultures. It should be noted that many of these observations were made on decompressed samples. While such results of ambient pressure experiments might extend our view of the deep biosphere, they are nonetheless biased towards surface pressures and likely do not reflect microbial processes at in situ pressure conditions. The limited number of piezophilic isolates also hinders our ability to investigate the effects of high pressure on metabolism and physiology. Piezophiles exhibit different strategies to cope with elevated pressure and maintain cell integrity over a wide range of pressure, including cell wall and lipid membrane biochemistry, intracellular salt content and osmolate regulation, specific high-pressure gene expressions, and macromolecule structural modulation. These unique piezophilic strategies have been identified via a limited number of high-pressure cultivation experiments, but there are diverse uncultured species within the deep biosphere that undoubtedly have known have unknown metabolic and physiological strategies. In the absence of additional isolates, these unknown functions have been probed with high-pressure experiments conducted on bulk samples from the deep biosphere. Among these more than 200 varied experiments, one general trend has emerged, is that the retrieval depth is broadly correlated with a positive response to elevated pressure. These results suggest that decompression and/or ambient pressure conditions are a challenge for subsurface organisms, and that incorporating elevated pressure conditions into sampling and experimental protocols could greatly improve experimental outcomes. Below, we review the sampling and cultivation techniques commonly adopted in marine subsurface biosphere research, the limitations inherent to these approaches and strategies to improve subsurface exploration through technology development and new collaborative models. Figure 1. Optimal temperature and pressure growth conditions for known piezophiles, light orange circles, and obligate piezophiles, red squares. Gray bars indicate the reported temperature and pressure growth ranges for each isolate. Collection of natural samples and isolates with sample origins at various steps and their response to elevated pressure compared to ambient pressure, 0.1 megapascals. Natural samples, solid, where metabolic activity was monitored, was reported to have either a negative, solid blue, neutral, solid gray, or positive, solid orange response to increased pressure compared to growth at 0.1 megapascals. Isolated piezophiles, dotted orange, are described here as having optimum growth pressures above 0.1 megapascal, and obligate piezophiles, dotted red, are those that require elevated pressures for growth and cannot grow at 0.1 megapascal. This figure is not an exhaustive list of all natural samples taken from the deep biosphere with reported pressure tolerances and does not include piezo-tolerant organisms, optimum growth pressures at 0.1 megapascal. Temperature and pressure data for isolates in A, B were collected from references in the supplementary table S2. Data for natural samples were extracted from the Picard and Daniel, 2013. How do we explore the piezosphere? How do we explore the piezosphere? Since the discovery of deep sea and subsurface microbial life, the development of advanced instrumentation for observing, retrieving, and manipulating samples have illuminated some of the key characteristics of the deep geosphere and biosphere. International collaboration and national efforts are the hallmarks of subsurface exploration, including drilling and submersibles. Similarly, large international programs support the research communities that explore the deep biosphere and improve our knowledge of the subsurface microbiome. Cf. ICOMM, International Census of the Marine Microbes. C-DEBI, Center for Dark Energy Biosphere Investigations, and DCO, Deep Carbon Observatory. Only 3 human expeditions have explored the deepest part of the ocean, Mariana Trench, roughly 11 kilometers, leaving 95% of the ocean unexplored directly, even though other HOVs, human-occupied vehicles, like the Shinkai 6500 and the Nautile that reach up to 6,500 meters allow for indirect exploration of nearly 98% of the ocean floor. Moreover, a veritable army of landers, ROVs, remotely operated vehicles, and AUVs, autonomous underwater vehicles, enable much of today's deep ocean exploration. While a new class of internet-operated vehicles, IOVs, and mobile platforms provide extended monitoring of both the seafloor and the water column. While drilling and exploration vehicles remain few Instruments to obtain subsurface samples are more numerous. Supplementary Table S1. The paltry list of isolated piezophiles reflects the difficulty of both retrieving and cultivating piezophiles from subsurface samples, because decompression affects cellular viability, and because most samples are decompressed during sample handling. It remains possible that most piezophilic strains elude current sampling and culturing techniques. Therefore, several pressure-retaining devices have been developed to enable sampling without decompression. Recently, Tamburini's team used a pressure-retaining sampler in the bathypelagic zone to measure prokaryote activity in situ pressure conditions and demonstrated that decompression suppresses both piezophilic metabolisms and water column community composition. A similar pressure-retaining sampler was developed as part of DCO's PRIME, Piezophile Research Instrumentation for Microbial Exploration, facility. The Pressurized Underwater Sample Handler System, PUSH, is similar to Bianchi's sampler, Bianchi et al., 1999, and uses a floating piston system to enable sampling, transport, transfer, and cultivation of high-pressure fluids without decompression. Accessing the deep marine biosphere and retrieving samples in situ pressures have proven a significant technological challenge for deep biosphere exploration. But these improvements can be amplified if subsequent laboratory studies, particularly cultivation experiments, maintain high-pressure conditions. It is particularly important to maintain in situ pressure conditions during sampling, transfer to the laboratory, and throughout cultivation and storage. In order to avoid decompression bias, when targeting novel deep biosphere microbes replicating in situ environmental conditions, i.e. pressure, temperature, pH, volatile composition, low nutrient concentrations, and subsampling without decompression are crucial aspects of the cultivation process. Since traditional laboratory culturing techniques are not easily adapted to high-pressure customized devices and protocols have been developed. Llanos and colleagues of the pioneers of high-pressure microbiology developed early high-pressure cultivation apparatus, and these have since been expanded. Cultivating piezophiles requires expertise in both high-pressure equipment, e.g. hydrostatic pumps, high-pressure vessels, as well as manipulating high-pressure cultures. Most of these high-pressure devices are batch reactors using a compressible reaction container in a static high-pressure vessel. Typically, a stoppered syringe containing inoculated growth medium will be pressurized in a hydrostatic pressure vessel. Unfortunately, this setup requires decompression during subsampling, potentially suppressing growth rates and cell viability. Variable volume reactors like the PUSH and others can overcome this limitation and have recently been used for batch cultivation without decompression, identifying two pressure-tolerant strains previously thought to be pressure sensitive. This technology has been extended to allow for high pressure transfer, a crucial step in replicating traditional liquid culture microbial cultivation and expanding the possibilities for high pressure isolation of enrichment cultures. A further improvement that better replicates natural geologic, environmental, and continuous flow-through systems. Janesh and colleagues first developed a high-pressure, 71 megapascal maximum, continuous culture system for low temperatures. Sauer et al. Designed a flow-through bioreactor, 120 degrees Celsius and 60 megapascal, that allowed for volatile equilibration of the culture medium, subsampling without decompression, and the inclusion of solid phase. Recently, in the Rogers lab, a similar continuous flow reactor was built that extended this pressure range to 100 megapascal and incorporated a module for sample fixation prior to decompression. Deep ocean sediments can also host a significant portion of the subsurface biosphere. However, retrieving deep ocean sediments and maintaining in situ pressures during cultivation and transfer of sediment present additional challenges. High-pressure sediments have been retrieved during drilling with pressure coring systems. But only the deep ISO bug system can transfer sediments under pressure for subsequent cultivation. Otherwise, most methods developed for high-pressure incubation and enrichment of sediments require decompression. Nonetheless, these devices can accommodate general batch cultivation, enhance volatile concentrations, and flow-through conditions, and have confirmed that microbial metabolic rates are faster at elevated pressures in the presence of increased volatile substrate concentrations. Given the extent of the subsurface microbiome contained in deep-sea sediments, the deleterious impact of decompression, and documented importance of elevated pressure on growth and metabolism, expansion, and improvement of high-pressure sediment manipulation remains ripe for high-impact technological developments. What are the limits? In the modern era of microbiology, the capabilities of Earth's extremophiles have far outpaced our ability to conceive of the bounds of habitability. For each perceived limit, temperature, pressure, pH, energy flux, etc., a novel and hard-to-imagine extremophile has surpassed expectations and extended the reach of potential habitable environments. In contrast, our technological capabilities have not kept pace with this ever-expanding habitable envelope. Even with the current known temperature and pressure limits of life, we have not yet explored most of the habitable subsurface. The primary limitations are the expense of drilling and oceanographic expeditions, together with technological limitations for sampling and sample manipulation. In the deepest parts of the ocean, pressures can reach 110 megapascals, and access to these environments is limited to only a few ROVs and landers. Recently, Bartlett and colleagues developed a modular lander capable of sampling these Hadal trenches at depths greater than 6 kilometers. While spatially limited, these regions nonetheless represent 45% of the depth range of the ocean. Sampling at these high pressures also poses technological challenges, as most samplers are designed to access meso and bathypelagic zones, 200 to 1,000 and 1,000 to 4,000 meters respectively, and only a few are designed to reach deeper zones. Access to subsurface crustal and sedimentary ecosystems is limited by drilling technology, and high-pressure retrieval of solid phase samples is rare. To our knowledge, only fluid samples have been collected from deep crustal aquifers at in situ pressures, and high-pressure sediment retrieval is pressure-limited. These latter examples highlight one of the more daunting challenges to understanding the deep biosphere, which is that the scarcity of samples. These latter examples highlight one of the more daunting challenges to understanding the deep biosphere, which is the scarcity of samples. Subsurface sampling is not only sparse, but often targets specific kinds of environments, resulting in a lack of coverage for the average deep subsurface. Therefore, it is difficult to extrapolate across the entire subsurface biosphere, leaving large errors on our estimates for subsurface biomass as well as metabolic, physiologic, and genetic diversity. Challenges, innovations, and opportunities. The last few decades have witnessed a dramatic increase in subsurface exploration and research, extending our access and our understanding of the deep biosphere. Nonetheless, sampling and analyses challenge remain. Here we focus on 4 areas of opportunity for innovation. Figure 2. First, sampling at in situ pressure conditions. 2. High pressure cultivation techniques. 3. Dissemination of high pressure skills amongst the community. And 4. Increasing participation and or incorporation of high-pressure sampling into deep-sea and drilling expeditions. We propose that community priorities going forward focus on deepening our knowledge of the deep biosphere and its role in the global biogeochemical cycles, particularly by addressing technology, development, and implementing novel management and collaborative models to increase community access to high-pressure samples and devices. Despite the ubiquity of prokaryotes within the deep biosphere, only a limited number have been cultivated, roughly 0.1%. Illustrative of this point is the distribution of the isolated piezophiles, with pressure maxima along the psychrophiles of the hadal zones and vent hyperthermophiles. The moderate temperature and pressure regimes, roughly 20 to 60 degrees Celsius and roughly 40 to 80 megapascals, lacks piezophilic isolates, despite the prevalence of such conditions within deep sea sediments. Similarly, the moderate pressure extent of the isolate hyperthermophilic piezophiles, where the optimum pressure is less than that of 80 megapascals, reflects the conditions of the most accessible fluids from mid-ocean ridge high-temperature vents. Ecosystems at higher pressures and more moderate temperatures, e.g. subduction zones, crust-off axis and spreading centers, are among the least accessed habitats of the subsurface. Exploring the full breadth of the piezosphere will require a new drilling and sampling techniques targeting high-pressure retrieval and manipulation of rock and sediment samples. More broadly, across the piezosphere, better cultivation methods that mimic in situ environmental conditions, i.e. temperature, pressure, chemical composition, could improve enrichment and isolation of novel piezophiles. Targeting several metabolic and physiological niches requires scaling up high-pressure cultivation either by vastly increasing the number of pressure vessels or developing new technologies for multiplexing techniques. The deep biosphere also hosts microbial communities with slow metabolic rates surviving on minimal energy sources. Traditional enrichment and isolation techniques favor fast growing under high energy conditions and are less likely to capture the slow pace of the deep biosphere microbiome. In situ or long-term incubations could help target the deep slow biosphere, but these require innovations for long-term incubations and high-pressure retrieval. While it is clear that every microbiological and molecular tool should be brought to bear to understand the deep biosphere, many of these need to be adapted to accommodate the elevated pressures of the subsurface. For example, monitoring metabolic rates during controlled incubations often relies on radio-labeled substrates, but adding radioactive material to pressure vessels introduces significant shipboard and laboratory safety concerns. Electrochemical monitoring of in situ chemistry during laboratory cultivation is also quite common in benchtop microbiology, but a more significant technological challenge when incubations are conducted at elevated pressures. Visualizing cells and microbial communities with high-pressure microscopy can be accomplished via specialized equipment, e.g. DACs, but it is hardly a routine as it is in the traditional microbiology workflow. Even the most basic methods like dilution to extinction isolation protocols, proteomic analyses, or compound-specific stable isotope measurements require either large numbers or larger volumes of high-pressure devices, limiting widespread use. Molecular techniques, which are often used to give more robust pictures of microbial diversity compared to cultivation-based approaches, have indeed revealed that subsurface microbial diversity is broader than represented by cultivated piezophiles. However, recent work suggests that even molecular techniques can suffer from decompression bias. Nonetheless, continued application of next-generation omics techniques could further elucidate the diversity and activity of the subsurface microbiome. and be used to improve culturing efforts. For example, geochip-based high-throughput metagenomics have highlighted metabolic functions within dynamic microbial communities. Furthermore, the combination of stable isotope probing, FISH-SIMS, and single-cell sorting could further elucidate the links between microbial diversity, environmental conditions, and ecosystem processes, and develop more robust models of the deep biosphere and its connection to long-term biogeochemical processes. Finally, we believe that sharing high-pressure expertise and equipment within the deep biosphere community and expanding the scope of high-pressure microbial research will advance our understanding of the deep biosphere. The number of facilities that can sample and manipulate subsurface samples at in situ pressures is considerably smaller than the number of research groups that regularly sample and study the deep subsurface. The barrier to entry, expense, and expertise into high-pressure techniques is high, but better coordination and new collaborative models can change this paradigm. The DCO, via the PRIME facility, supported the purchase of several push samplers to share within the community, with the goal of isolating and characterizing more piezophilic microorganisms. However, equipment sharing will only be successful if it is accompanied by the requisite personnel and expertise. Therefore, new collaborative models that include equipment, personnel, and funding needed to be developed to keep community-wide facilities successful and sustainable. The Earth's deep biosphere not only represents a crucial segment of the global biosphere and modern biogeochemical cycles, but it also serves as a window into Earth's habitable environments throughout its history. Furthermore, as the exploration for life beyond Earth shifts to other solar system bodies, including the subsurface of Mars or the subsurface ocean worlds of Europa, Enceladus, and other moons, understanding how these environments can foster life becomes crucial to this exploration. Therefore, the technology developed to explore Earth's modern deep biosphere will eventually become a key component of missions to these bodies to search for signatures of extant extraterrestrial life or its remnant biosignatures.